Unit 8, page 3. How does DNA replication and protein synthesis occur? Do now. See if you can write down what the central dogma of DNA is. Take a minute, pause the video here, press play when you're ready to check your answer. Okay, so it looks like you're ready to check. You should have DNA, RNA, protein. Now we've added in here cells and body, but you should always remember DNA, RNA, protein. DNA is going to have the information to build proteins. That's how it works. We call these genes. So genes are basically pieces of DNA that encode for a protein. So here we have a piece of DNA. It then encodes for a protein and allows us to make our cells. And from our cells, we can have multicellular organs to make the body. And of course, DNA gets all the glory, but it's really the proteins that are doing all the work. Replication of DNA. Before a cell can divide by mitosis or meiosis, remember mitosis is what type of cell division? That's right, asexual. And what do you use mitosis for? Good, for growth and repair. Meiosis, what type of cell division is that? Good, sexual reproduction. And what are the only things that meiosis does? Good, it makes gametes or sex cells. It makes only sperm or egg. So before a cell can divide by any type of cell division, it must first make a copy of its chromosomes. The DNA is copied in a process called DNA replication. In DNA replication, the strands are going to separate when an enzyme called helicase is actually going to unzip the genes or unzip the DNA. The DNA opens, so we can see here how the blue DNA, it opens up so that each of the strands are exposed. So the strands separate when the enzyme cuts the DNA open and uses each strand as a template or a pattern to assemble the new strands. So as you remember with the base pairing rules, A is going to pair with T, C pairs with G, G pairs with C, and this now is an exact copy of the original strand. And we can see here we get two new copies that are identical to the parent molecule of DNA. How do proteins do all the work? Good question. So proteins are going to run living organisms. They do this through the use of enzymes, which control all chemical reactions in living organisms, and they also do this through structure. So all living organisms are built out of proteins. Things like collagen, keratin, these are all proteins that make up our body. Cell organization. DNA, again, is found in the nucleus. So we have here our cell, the cytoplasm, and we're going to draw our little nucleus here. Inside we have the DNA, which again are the instructions for making proteins. We want to keep it there, we want to keep it protected. So essentially think about the nucleus as locking that DNA in the vault. Proteins are chains of amino acids. So we have here some amino acids that are joined together. They are folded together to form functional shapes, whether that be an enzyme with an active site or just a structural protein that makes up a part of our body. And they're made by a protein factory, if you will, in the cytoplasm. And we call this the, ribosome. So the protein factory are the ribosomes. They're in charge of building the proteins, and they do this by getting specific instruction from the DNA about which amino acids, how many, and what order. So how does the DNA pass the information about what type of protein to make to the ribosome? We need to get the DNA gene information from the nucleus to the cytoplasm. So we need a copy of the DNA. We do this by actually making messenger RNA. So we have the DNA, goes to messenger RNA, and then it's translated into protein, and then we get whatever trait it is that the DNA encoded for. So how does this happen? Well, first let's think about the DNA versus the RNA. So you can see just looking at the pictures right away, what do you notice about the DNA versus the RNA? Good. if you notice the fact that the RNA here is single-stranded and the DNA is actually double-stranded, that's one of the primary differences. But also in the name itself, we have DNA and we have RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. DNA is going to have a deoxyribose sugar on this blue band with the phosphate, whereas RNA on its blue band here is going to have phosphate and a ribose sugar on its backbone. They're both going to contain nitrogenous bases. DNA, as you remember, guanine, cytosine, adenine, and thymine, with T pairing with A and C pairing with G. RNA also has these same nitrogenous bases on its nucleotides. However, you'll notice one difference. We have guanine, cytosine, adenine, and instead of thymine, we have U, which stands for uracil. U is going to pair with A, because it's replacing the T, and C is going to pair with G. As you already pointed out, DNA is double-stranded, whereas RNA is single-stranded. Now, transcription. This is the process of making mRNA from DNA. So we have our double-stranded DNA. We're going to use the process of transcription to make our single-stranded mRNA. We use DNA strand as a template or a pattern. We're going to pair the complementary bases. We're going to do this by pairing a U in the mRNA anytime the DNA has an A, and a G with a C. This is going to take an enzyme called RNA polymerase. Remember, a polymer means many, and anything that ends in ASE stands for an enzyme. So let's look at this a little closer. Matching the bases of DNA and RNA. So again, this double-stranded DNA has to be unzipped. Then RNA polymerase is going to match the bases. So match the RNA bases to the DNA bases on one of the DNA strands. So let's take a look at how this matches. So T is going to pair with, so again, the U instead of T is matched with the A. So let's see how well you can do this. Pause the video here and try it. Press play when you're ready to check. Okay, let's take a look. So we'll see the complementary base pairs. T would go with A goes with U, C goes with G, G goes with C, C goes with G, A goes with U, and so on. So it's just the same pattern over and over again. Whenever you see up here, the complementary strand is below. Now this is going to travel to the ribosome. And the ribosome is going to make a chain of amino acids which forms a protein. So again, nucleus has the DNA, we make the mRNA, that mRNA goes to the ribosome, and our ribosome is going to make the chain of amino acids which forms a protein. Our protein then is our trait. How does mRNA code for proteins? Again, mRNA leaves the nucleus, mRNA goes to the ribosome in the cytoplasm. Proteins are built from instructions on the mRNA. So we're going to use these instructions. You'll see here ACC, AUG, those are the instructions to get our amino acid. But how does it do this? How does mRNA code for proteins? Well, again, we have our DNA. It makes our mRNA. And now what happens at the ribosome? Well, at the ribosome, we're going to have proteins joined together in a specific order based on this code. How can you code for 20 amino acids with only 4 bases? Well, mRNA codes for the proteins in triplets. This means that for every 3 mRNA bases, you get 1 amino acid. So the first three, the next three, and so on. We call this a codon. A codon is a block of 3 mRNA bases. Now, we have a chart that we're going to be using in class to practice going from the mRNA codon to figuring out which protein. So if we take a look at this chart, you can see here that the first bases are listed here, the second are here, and the third are here. So if I have something that is AUG, I look on the first part here, I see A, I see U is the first column, and I see G, methionine or start. mRNA code is for all life. The strongest support for common origin of all life is the fact that we all use the same mRNA codons. The code has duplicates, so you'll notice the brackets that several codes encode for one type of amino acid. And this is essentially mutation insurance. So if that last letter in the codon changes, it's still going to come out with an OK protein. We do have a start code, as well as a couple stop codes that will tell the ribosome to stop translating the protein. So how are the codons matched to the amino acids? This is with the help of another RNA molecule known as tRNA. tRNA is essentially going to bring over or transfer the amino acid. It does this with what we call an anticodon that pairs with the mRNA. An anticodon is a block of 3 tRNA bases. The mRNA to protein we call translation. This is working instructions of mRNA. The reader is the ribosome, and the transporter is the transfer RNA. So we'll see here the transfer RNA brings this over, then it detaches. Meanwhile, the amino acids are slowly being linked together. So from gene to protein to review, we go from DNA, mRNA. This is called transcription. mRNA to protein happens at the ribosome. This is called translation. Proteins are formed by the joining of amino acids. Remember, the order and the number of amino acids are really determined by the DNA. The transfer RNA is going to assist the ribosome in bringing the correct amino acid over. Okay, so to review, we have the nucleus, which contains our DNA. We make from the DNA mRNA in the process of transcription. mRNA is going to travel out of the nucleus to the ribosome and then undergo the process of translation. The tRNA is going to bring over the appropriate amino acid to match the complementary strand of the mRNA. And once these amino acids are joined, we're going to make a protein, which determines traits. So again, DNA is transcribed into mRNA, mRNA is translated into protein, and proteins determine our traits. Here's another look at the same process. Again, it's important to realize that the DNA code, the reason why it encodes for our traits, is because it determines our proteins. Remember to write a question about this unit, write 2 of them on your paper, and write the answers.